Hey guys, CRISPR here. So for our first story of the day, a team of entomologists at Penn State have developed a technology to edit genes in all arthropods, which is currently unable to be done with CRISPR. This technology is called remote control, which stands for receptor-mediated ovary transduction of cargo. Remote control builds on the CRISPR-Cas9 system and can deliver gene editing cargo to a targeted portion of the arthropod genome by easy injection into female arthropods during egg development. This is very interesting, and I hope the scientists can apply it to other uses as well. For the next story, scientists at the University of Texas, Austin, have developed a sort of predictor for the off-target effects of CRISPR. The method involves testing the CRISPR molecule against the whole genome instead of just the target area. The technology is called CHAMP, which stands for Chip Hybridized Affinity Mapping Platform. Hopefully, this allows scientists to ensure that the CRISPR edits are safer and more effective. Also, remember that study that was done a few weeks ago hoping to cure blind mice with CRISPR? That study that supposedly revealed many off-target effects in CRISPR and also caused the stocks of companies like CRISPR Therapeutics to drop in price? Yeah, well, there's evidence that the supposed mutations from that study might not have been related to CRISPR. A Harvard University-led team has suggested that because the mice were used in the experiment were closely related, the mutations could be genetic instead of being off-target mutations caused by CRISPR. Further evidence towards this theory is that the mutations were not found near the gene editing site. This is good news because it shows that CRISPR is safe after all. And for our final story of the day, we have a big one. A new method of gene editing has been developed by researchers from Johns Hopkins University, Rutgers University, Harvard, and the University of Trento in Italy. This method is called LASSO, which stands for Long Adapter Single-Stranded Oligonucleotide. It can capture and clone long sequences of DNA fragments. It can read more than 1,000 base pairs, whereas next-gen sequencing, like CRISPR, can only read about 100 base pairs. Because of this, it might actually replace CRISPR as the best gene editing tool. I might have to change my name to Lasso Man. Anyway, that's all for today. See you next time. This is CRISPR Man signing off.